is Trisha with Insectopia, and we are here to really dive into aquatic biomonitoring, especially with our friends, the benthic macroinvertebrates, or that's at least what we call them. All of those little friends who are living in and around the water, and if we collect them, we can actually determine water quality. So we're going to be going over that. I know that Avea is here, and I am happy to see her. I hope everyone else is happy to hang out. I was thinking I might just tell you kind of the order in which we would go over things today. There is so much to biomonitoring. It's hard to fit it into one live stream, but we're just going to do the best that we can. And it is all going to be useful. So, very good. We've got some habitat assessment, so not even just kind of before you get into the water and before you're collecting your aquatics, just being able to look at an aquatic system and know what a high quality system looks like and what a low quality system looks like. We're going to be using this book, the Rapid Bioassessment Protocols for Use in Wadable Streams and Rivers. It is actually by the Environmental Protection Agency. It's by the EPA. And it's just a large, essentially a data book for... this is what I'm going to be, we're going to work on. We're not going to cover everything in here because this calls for periphyton and fish and stuff, but we're going to be looking at some of the habitat assessment stuff that they use. We're going to talk about the different jobs that says whether or not your aquatic system is... whether your aquatic system is healthy or whether there's all types of pollutants in it. You can read all types of stuff into it. All right, let's get going. I figured we would come on over here and use my table camera for a little bit because this first little bit we're not going to have the microscope. All right, so in the back, and I think what I'm going to do is, if you want a scanned PDF of some of these data sheets, I can scan them and email them to you. So just go ahead and email me if you would like the data sheets that we're going to be looking at today. So a lot of times when we are looking at a river, you've got all of the basic stuff we take care of: the name, the location, who's doing the paperwork. And when we are recording environmental, we're doing habitat assessments, we are a lot of times going to be drawing a stream map, which is we're going to make sure that we put in things like fallen trees and riffles and any of the pools or the quick shallow areas, those types of things. Any pipes that lead into the water. And the reason we sketch out those maps is because you want to make sure that over the course of time, our rivers are changing, the way we want them to, and they're going in a more healthy direction. I don't want to go over all of this. What I do want to talk about are what we're looking at in aquatic systems and where the insects are going to live and why they prefer this. So we call this the available cover or Epiphonal Substrate. So when you're looking at, this is what is above the river, right? Because if you think about, I'm going to move myself just a little bit so I'm not in front of this so much. if you're thinking about when you're looking at your water quality and you're looking at your stream, you want to be considering what's above the river too, because all of these leaves are going to be falling into the river, and it's going to create more habitats, and it's going to create kind of a healthier environment. So this is what we would consider optimal range. And then over here you've got a poor stream where, yes, it's got beautiful grasses and all the way up to the edge, but if you look above, you don't have those same overhanging trees and limbs and those types of things that are actually beneficial for our stream. And so for all of these assessments, you can kind of put it into four categories, whether it's optimal, suboptimal, marginal, or poor. Another thing we're going to be looking for when we're looking for areas for healthy aquatic invertebrate homes, or for healthy even fish. Your rocks, you don't want them to be embedded in soil and embedded in the silt. You actually would prefer to have your embeddedness be kind of lower, where you can see the rocks, and our insects can scurry kind of almost underneath the rocks or walk around the rocks. When you're looking at a poor stream, there's going to be silt and sticky mud and those types of things all over the stones. And you can give yourself, theoretically, 0 to 25%, all the way to 75 to 100% embedded in the silt. Ivea would like the data sheets, so perfect. I get your emails all the time, so I look forward to getting that one, and I'll send you your data sheets. And just let me know if you have any questions about what we're looking at in habitat assessment as we go through. Is your habitat that you're looking at, is it a stream where it's a moving system, or is it more like a pond or a lake? So with this one, we're mostly talking about, they're saying the pool substrate characterization, but they're talking about what is the substrate or the bottom of the river where you have pools or standing water. So you can have a pool where the water isn't running very quickly. You can have a riffle where the water is moving super, super quickly and you have it kind of turning over rocks. Or you can have just a run where the water is moving quickly in one direction and it's not sitting still. So we're looking at kind of what's on the bottom of the pool regions, and what you want is a mixture of gravel and like thick you want a mixture of like gravel and more solid substrates, whereas you don't really want something that's like a bedrock with no root vegetation, no like roots on the edges, and no, if it's all mud and all clay, bugs don't really like that, right? And where you don't have insects, you also generally aren't going to have fish, because you need insects for the fish food. And a lot of times, especially in like those embedded rocks, fish are going to lay their eggs amongst the rocks. There are some species that lay their eggs amongst the rocks, and if the rocks are too embedded, the fish just won't be there. We like to call these the velocity depth combinations, and I was almost got into this, but I knew we were going to be getting there. So there are 4 velocity depth combinations. You can be quick or slow, and you can be shallow or deep. And those are all 4 of them, and you can intermix them. So you've got slow deep, slow shallow, quick deep, and quick shallow. And ideally, you want all 4 of those types within within like a 100-meter range of our streams or our rivers. All right. We don't need to do that one as much. So I wanted to look at a couple of those in the beginning were the ones that I really wanted to talk about. If we want to look at something like this, you can talk about kind of channel alterations where obviously a natural stream or a natural creek is going to be ebbing and flowing and is going to be flowing in this S-shaped pattern, whereas something that was cut by people or was obviously, I think this is in between two farmland, two planted regions. That's what we would consider poor, right? So the water isn't going to, it's all in one direction, it's all channeled, that's no good. And over time, if you let this, if you just left this one alone and it didn't have any human interaction, it could go back to a meandering stream just naturally through what do we call that? Erosion of the edges. When I mentioned riffles earlier, I mentioned that they were water moving very quickly over rocky regions. And so we've got some riffles here. I think that you know most of those. Very good. Aha. So the other thing that we do talk about is the Riparian Edge. So this edge of the stream, what's growing on the edges of the streams? Obviously, a stream that is mowed all the way to the edge, that's not going to be a natural stream edge, and therefore you're not going to, that's kind of on the poor side of things. You really, really want to leave a natural edge so that the plants and the trees and those types of things can kind of lean over. There was a number that I wanted to look for really quick. I believe it's like 16. Where is it? I'm not going to find it. There is a recommended distance for the Riparian Edge, and if you're curious about that, I can find it. I don't remember what the number is right now. All right, so that's habitat assessment. And if you go through all of those habitat assessments and you give them a score, on a score they actually do on these assessment sheets, they do 0 to 20. And you can come up with a total final score at the bottom and then compare it to other stream systems, or you can compare it to itself. So if you do this over the course of time, you can see how your stream is changing, ebbing and flowing, and those types of things. That's awesome. All right, so that is our first little thing on habitat assessments. This one. Lost my page. Here we go. There we go. I believe that a number of you have already heard some of these. So I just want to throw that up there. I was trying to get my paper and the book to fit at the same time, but I don't know if it's going to happen. So our book gives a number of functional feeding groups. Some of these I personally believe you can kind of smush together. But like the scrapers and some of our collectors are the same, or the collector, there's a couple of them that are a little bit odd, but I can tell you what all of these jobs are, and then we can talk about them. So, obviously, our predators, ooh, you know what? I have a predator under the microscope, so we might as well check it out. The predator underneath the microscope that I have right now is a dragonfly nymph. They have this very, I love that. They have this, like, large head. They have a very, like, unique body shape. And they actually have this bottom jaw that has the ability to shoot out, grab its prey, and then pull its prey to its face. So it has this kind of jaw that reaches and grabs. I also have another dragonfly, but that's one of our predators. We can have what we call a gatherer or collector. A lot of gatherers or collectors, these are insects that are going to be crawling around on the bottom of on the benthos, on the bottom of the stream, and they're going to be looking for dead plant material or things that are breaking down, decomposing, so that they can collect and eat it. So those are our collector gatherers. A lot of times they're going to be on the ground, but not as much on the rocks, because we also have our scrapers, and scrapers are normally on rocks rather than just on the substrate. So they're going to, there's this really cute species of mayfly. Oh, I wonder if I have him right here. I think I do. Aha! I do have him! These aren't in their final vials yet, but I can show you this mayfly. He's in the same container as the water penny from last Thursday. I don't see my forceps anywhere. This is our friend a mayfly nymph, and I've always loved their heads because, or at least this family, I believe it's Heptageneidae, but we're going to be getting there a little bit later. So then I will have that information. This is a mayfly nymph, and its head is really, really flat, and it's a scraper. So it sits like this right across the rock, and it scrapes all of that algae and anything that's kind of growing on the rock, and that's what it's going to eat. And when we're identifying mayfly nymphs, our rule of thumb is that mayfly nymphs have 3 tails, right here. All right, so that's how we, there are also damselfly nymphs that have 3 tails. So those are I guess those are the 2. Mayfly nymph tails are thin and long, kind of like hairs, whereas a damselfly nymph, I actually have one here too. So this is our damselfly nymph, and it has the same head type that a dragonfly nymph would have. It has these really big eyes, these kind of short, these filiform or straight antenna. It also has that bottom jaw that can shoot out like a dragonfly. Both dragonflies and damselflies have that really cool bottom jaw. But if we look back here, it does have 3 tails, except damselfly tails are more like a leaf or a feather. They're flat and wide, whereas mayfly nymph tails are long and thin. All right, so they both have 3 tails, but they're very different, and you can very much tell those guys apart by that. Do larval damselflies look similar to larval dragonflies? I hope that answered your question. We are going to also be looking at a couple of other species of we're also going to look at, I guess, another picture or 2 of damselfly nymphs. All right, so that was we just talked about the scraper. There are also insects that we call shredders. So shredders are going to be taking large pieces of organic material, and they're going to be breaking it down into smaller pieces of organic material. And if you spend enough time with macroinvertebrate people or biomonitoring people, you'll hear the words CPOM and FPOM. And they are, well, we'll talk about them one at a time. So CPOM means coarse particulate. It's coarse particulate and then organic material. When we're talking about CPOM in the water, that's any of the big pieces of organic material. So big leaves, big sticks, anything that is organic material coming out from outside of the water, entering our water, that's CPOM. And our shredders are insects that really like to spend their time inside of like leaf bundles or over on the inside of logs, and they break big pieces into smaller pieces, and they eat it while they're doing it too. So they're breaking all of these big pieces down into small pieces, and if you are I don't know, if you're looking for a good just color picture guide to aquatic invertebrates, I really like this book, Freshwater Invertebrates of North America, because it has basic color images of pretty much everything you're gonna find. And it's built for people, it's built for the average person, so you should be able to pick it up and be able to read it and understand it and use it right away. So I wanted to show you, this is going to be one of our really common guys right here. We were just mentioning shredders. They like to spend their time in leaf bundles. And this is a cranefly grub. And craneflies are huge shredders in aquatic systems. You find them in streams. You find them in lakes. You can find them all over North America. So they look like giant maggots. And sometimes they can be really big. They're actually kind of awesome when you can find them. And they're really big this time of the year. January, February, right after all of those leaves fall and they really start breaking down the craneflies, they just go to town. And you'll even find them if you break through the ice and you find a little leaf pack. You should be able to find craneflies. They are mildly sensitive species. You would kind of put them right there in the middle. So, we'll talk about that shortly. So the damselfly nymphs are also predators. Yes, damselfly and dragonfly nymphs are both predators. Alright, so that was shredders. We also have what we call filter collectors. And there are some, well, I want to show you these guys first. We're going to look at these guys. 1 minute. So I am just leaving my specimens in the vials that they are in, but we can see the friend, oops. We can see our friend and the home that it built. Oh, come on. There we go. All right, so I'm not sure where limnophilids fit on their, like, exactly what their job is, because caddisflies can be shredders. They can be filter collectors. And they can be collector-gatherers, just depending on what family you're looking at. But this is a little caddisfly, and caddisflies generally are going to be making themselves cases. And to me, they're taking big pieces of leaves and they're cutting them into pieces to make these little homes here. So they do have little silk glands up in the front of their face, and what they do is they will cut these these limnophilids, they cut these perfectly circular discs out of leaves, and they silk them together, and they make themselves this little case. Generally, they are not going to be leaving the case, but when you collect them and put them into ethanol, you want the specimen separate from its case, just so that you can ID it by its characters on its legs and its heads and those types of things. But you also want to keep the case because sometimes the case helps you identify them to family by what they look like or what they're made, the materials that they're made out of. Each of the families kind of has their own unique way of making their cases. And those are caddisflies. All right. Other than our caddisflies, I'm gonna leave it on there when we go to the next one. Oh. It overfocused. Where'd the bubble? No, we don't want the bubble. I wish I had my aquatic collection from the one I turned in from school. That would have been awesome. I don't have that one anymore. Alright, so the last of the jobs that we haven't covered are the filterers. And for me, a lot of times I'm seeing in habitats that are disturbed a lot of times, you end up with a lot of filterers. We'll talk about that in a minute. But filterers are going to be any of those insects that are actually filtering stuff out of the water. There is a group, a family of flies. We call them the black flies, and they are really, really commonly spoken about when we talk about filtering because they have a head up here in the front. They've got a kind of a narrower thorax, but then their abdomen kind of gets really wide. They get called out for looking like bowling pins. And that's how you identify black fly larvae, is because you look at the rocks, and the rocks will have little bowling pins on them, and they'll be kind of waving back and forth just a little bit. But the really cool thing is that up on the top of their heads, they have what we call labial fins. So instead of having labial palps like those mouth fingers, they are adapted to look more like this. And they open and close in the water there, like this, and they will fan out, and they filter all of the, what we would call FPOM, which is the fine particulate organic material. So they take all of the little itty bitty pieces of organic material out of the water, and what they'll do is they'll take it back to their mouth parts and they run their fans through their mouths to clean the organic material off, and they do it again. And it's really, really super cute. So if you ever have the chance, you can look up black fly larvae on YouTube, and I'm sure there's a video of them out there somewhere. I haven't taken a video of that yet. And I guess I should give you this. The black fly larvae, they're in the family Simuliidae. Okay. Alright, so those are jobs, and habitat determination. There is a little bit of the river continuum I'd like to discuss because I kind of love the river continuum concept, but along with that is if you have an idea of this concept in your head as you are looking at waterways, you'll have an idea before you even step into the water what you would expect to find. So We call this the RCC, or the River Continuum concept, and the idea is that streams are ranked in orders. There are stream orders, and you can have a first order stream that is a very, very thin, narrow stream. I always imagine first order streams like the trickling streams up in the mountains. They're where water starts. All right, that's the very, very first order stream. And when you have a first order stream that meets another first order stream, you have a second order stream. And that one is just a little bit wider. It's got a little bit more potential. It's probably still in the forest somewhere. It definitely has a lot of shade cover. It definitely has branches over top of it. It's probably not, there's not very many people out there. So I'm going to give myself another one. We've got another first order stream and another first order stream. And then we have the second order stream, where a second order stream and a second order stream combine, you end up with a third order stream. And so forth and so on. So you can go all the way up. I believe you can get to like 8 or maybe 8. Maybe up to 10, but probably not much higher than that. And as you go up in order, the different types of insects that you're going to find changes because of the amount of organic material that's entering the water and the the needs of the environment. So in our, what we would consider our base order streams, those would probably be about, in my head, it's about 0 to 4. If you were to look in one of these, if you were to look into one of these, you can give yourself almost this little pie chart. And it doesn't matter if you are 0 to 4, 5 to 6, 7 to 10, 7 to 8. I'm going to have to look up stream orders, but you always have about 10% predators. So these are our little predators. These are our dragonflies, our damselflies. Some caddisflies can be predators. A good number of beetles, like predaceous diving beetles. Yeah, so there are all types of predators, but there's only about 10%. And no matter where you are in a system, that should be your expectation. The predator may change based on if it's a really, really thin stream or a big one, but that's what you should expect. I want to find this in my book. Because I had just looked this up, and I can describe it to you, but I would like to have the numbers correct. All right, we're just going to go for it. All right, so from here, we are going to have a small percentage. I'm going to block that one out essentially for everybody else and then cut these two in half, 40 and 40. I believe this is about 40% and 40%. So you've got 40, 40, and then 10, 10 of kind of everyone else. Now, up here in this 40, 40, we're going to be splitting between our shredders, because shredders are going to be very, very important in low order streams. You pretty much don't find shredders in high order streams because you don't have as much of that big, coarse organic material, right? So you don't have those fallen trees and those branches and big rivers like the Delaware River or the Mississippi River, right? Those are huge, wide rivers. Whereas in these low orders, you have about 40% of them that are shredders. It's their job to just consistently consistently break up all of those leaves and stuff that are falling into the water. Then you also are going to have that for our collectors and our gatherers. And then you've got those collectors and gatherers. And we're just going to do low order and high order just so that it doesn't get too confusing. So I'd like to go do the high order, which I mentioned is always going to have that 10% predators, and then you're still going to have about 10% of everybody else. The thing is that you need collectors and gatherers still, because all of those, there is some material falling into the water, and so the collectors and gatherers have the ability, they're very mobile, and so they're going to be out there. But now, instead of having so many shredders that are breaking all of these big organic materials, you have the individuals that need to break down the fine particulate organic materials, the FPOM. And so those are going to be our filterers. So you switch from having things like crane flies and caddis flies and insects that are going to be breaking down big pieces to things like scuds, which are freshwater shrimp, or like isopods. They're sow bugs. They're like aquatic roly-polies, filters, and black fly larvae, which is a filterer, right? So as our streams get wider and wider, that's what we kind of expect to see. There is an in-between ground in there. I don't remember what the percentages are, so I just wanted to kind of share this concept with you. And that is a concept that, you know, you can't take the numbers to the bank every single time, but it's a general idea of like, hey, this is what you're looking at, this is what you should expect. And after doing habitat assessment and knowing what what the jobs are and what you might expect, then all you need to do is to be able to ID our bugs now, right? All right. Let's do this. Now, I think how I would like to go through some of these specimens is I just want to kind of share with you some of my notes and things that I have learned over the course of time. I will admit that some of these images I didn't expect to be using on a live stream, so not all of the images are mine. But I think that these will be good to share, especially for identification purposes and all of those things. Now, all right. So there was one thing that I wanted to look at, and I was trying to decide if we were going to do it first or secondarily. So there are 3 different types of aquatic system. You can kind of group insects into 3 groups. We can call them the sensitive groups, and these groups are the insects that the sensitive insects are the ones that are sensitive to pollution. So they're the ones that we really, really, really want in the streams. We want to find these guys. We want to find them in large numbers. We want to find multiple species of all of these families. So that's what we're looking at for a sensitive. Then you have what we consider moderately tolerant, and I'm just going to put mod tolerant. So these are moderately tolerant insects. These are insects that you like to see. They're great to see. They are not super duper sensitive, but if you have a large number of them and a high diversity of them, that's really what we're looking for. And then finally, you have what we would consider the tolerant insects. And the tolerant insects, those are insects that when you see them, it doesn't mean that the water quality is bad. It just means that like, they could live in they could live in really gross water too. So it's not telling you much about how polluted the water system is, because they could live in pretty much everything. If you have an aquatic system where you find lots of all three, that's a decent system. If you're finding only the tolerant insects and you don't find anything sensitive, overall, that's not going to be very good. That's telling you that there might be something wrong, or there might be a problem with your water system. All right, so as we're going through these insects, we're going to be classifying them as sensitive, moderately tolerant, and then tolerant. If you were looking in a book, like the aquatics, like the aquatics book, if you have this one, it's a big one. It's a really, really awesome one if you are really, really going to dive into this. If you have this book in the charts, the tolerance numbers are actually numbers on a scale from 0 to 10. So we would consider anything from a 0 to 3 sensitive, a 4 to 7 moderately sensitive, or moderately tolerant, and then an 8 to a 10 tolerant. So if you ever are running into tolerance numbers and you find numbers rather than words, that's what you're looking at. Avea, are you still following? Is everything good? I know I haven't heard you ask very many questions. Are you just kind of trying to take in some of our knowledge here? I just want to make sure we are we're all good. So, we have our Dobson flies. Now, Dobson flies you can find in all different types of habitats. You can find them amongst riffles, kind of underneath rocks. You can also find them... Oh, I'm sorry you have to keep reloading, but okay, I'm glad that you're still here and that we're still good. I just wanted to make sure. So, our Dobson flies are what I would consider what are considered moderately tolerant. When you give a generalization to an entire family, it's a little bit different, because as you go and you identify further, if you identify down to genus or if you identify down to species, there are small differences in the tolerance numbers of individuals. So you can generalize, but just keep in mind that there are some individuals that might break the rules. The rules are made for breaking. And I'm going to skip some of these that you're likely not going to see. But I love these guys, and I've got a story about them, so I'm going to share them with you. So this is an aquatic caterpillar. All right, the aquatic caterpillar has you see all of these little strands. Those are actually gills, and they do make themselves a little case, and they'll cut a little disc out of a green leaf, and they'll silk it from the top to the bottom, and then they live in it, just like a caddisfly makes its own case. These little caterpillars will make their cases, and they live underwater. Now, I caught one time, and it was the cutest thing on the planet. I absolutely loved it. And I thought it was a caddisfly, because it was just a case with a little head poking out. Except then, the head poked out the other side. And caddisflies only have an opening on one side of their home. And I was like, what is happening? Because I guess this little caterpillar kept turning around in its home and poking his head out of either side, and it was really super cute. So I loved that. And yes, aquatic caterpillars. 5.5 pages of notes. Oh, perfect. I'm happily not overloading you, I guess. You'll be able to kind of to like center yourself with the information and then come back at it. My caterpillars, I would consider I think that they are sensitive. I have only seen them, I've collected in a lot of places. I've only seen them in very, very clean waters. Yeah, at least these guys, the pyralids and the crambids that make their own cases, I would say that they're sensitive. I would say the noctuid, these guys are really, really big. They have a difference in their crochet pattern, but you find these at the base of rotten cattails. That's where you can find them. If you see a whole bunch of cattails on this edge of the road, you can pull them out, and at the very base where it's kind of rotting and black, if you break it open, you might be able to find these caterpillars. And I would say that those are moderately tolerant. Oh, good! Yay, perfect! That's what I like to hear. So we did these recently. We did water pennies. Admittedly, there are... Oops, that's a bad word. Admittedly, they are, there's two different genera, and look, we did the Cephenis because it had an entire margin, whereas the Ectopria, the other genus, has a serrate margin, and they look like a pill bug. That's what that says. Alright, let's see. How about this? If you have questions about specific families of sensitivity, some of these beetles I'm not going to know, but we can look them up later if we would like. So these are Riffle Beetles. They have really long legs. They also have really long tarsal claws. That's how we're going to be able to identify them. And when you find them in riffles. You find them amongst the rocks. They do swim a little bit, but a lot of times the beetles you'll find kind of bouncing around from rock to rock. I don't think that they jump. They don't have jumping legs. But sometimes they bop around. Yeah, that's what they do. So those are our Riffle Beetles. I would consider them they're moderately tolerant. Our Whirligig Beetles. These guys I would almost consider not tolerant or tolerant in the middle. I would put them on an edge somewhere. These are Whirligig Beetles. Now, you'll see these on the top of the water, and when I'm thinking about sensitivity, a lot of times it has to do with characteristics like what they're eating and then how they're breathing in the water. Whirligig beetles can carry their own water bubble from the surface. So they don't have to rely on oxygen underwater. And when we're talking about like high quality water needs to have oxygen in the water. It needs to be highly oxygenated. So we like to look at insects that need highly oxygenated water. The Whirligig beetles are not that. They can just grab their air bubble from the surface and then dive down and swim around. The fun thing about Whirligig beetles is that they have 4 eyes. They have a compound eye above the water, and they have a compound eye below the water. So if you were identifying these to family, that's how you would do it. And then you have our water scavenger beetles, the Dytiscids. A lot of times, yeah, that's fair. A lot of times, these notes I meant were, these are some of my notes from college, so admittedly, you know, some of the comments are funny. Water scavenger beetles have a differentiation in their antenna and their palps. I think we've looked at this before. They've got really long antenna, they have really short palps. And when we're looking at the other, there's another species of diving beetle that can look very similar, but these are flipped. So when we're talking about water scavengers, long antenna, short palpi. This is the other one. And this is what I wanted to show you. So if we're looking at this, these that are coming out, those are not actually the antenna. Those are the palps. that our beetle would use to kind of collect its food. And its antennae are actually really, really short and stubby. They're actually so small you can't see them in this image. But that's how we're gonna tell those guys, what we would call the predaceous diving beetles, from our water scavengers. These guys also have really, really, really awesome larvae. But these guys are predators. So predaceous diving beetles are predators as grubs and as adults. So you can expect to find them in that 10% predator range. But they also do not require highly oxygenated water. So these diving beetles are regularly found in high-order rivers as the predator in high-order rivers, because they can breathe the oxygen from the surface as grubs and as adults. Oh, these are so much fun! Okay, I'm going to show you this really quick, because I haven't shared this story in a long time, and I should have proofread these before I shared them with the world. That's okay. So, I don't know. So anyway, we've got these leaf beetles, and these leaf beetles are really awesome because they are really kind of short and fat, but they also can be found at the base of lily pads, because they use these they use these essentially spines on the end of their abdomen, and they can pierce a plant, and at the end of these spines they have spiracles so they can breathe, and instead of breathing the oxygen in the water, they can breathe the oxygen through a lily pad. They can kind of breathe through the lily pad. So that is one adaptation that actually makes this animal pretty tolerant. So if you find them, it doesn't say that the water is unhealthy. It just means that, hey, you've got lily pads around and these are common species. You're trying to figure out what the where to find the orders of streams. So, if you look up river continuum concept, you should be able to find that. A lot of times, the stream order is not only... If you don't have the ability to follow the river all the way back to its base, if it's already a pretty wide river, a lot of times you can measure the width of the river, and then there's a stream order designation for the measurement of the width of the river. I might have that. I have been trying to prep for teaching you guys about this for a while, so I have seen like all of my aquatic biomonitoring stuff recently. So it's it's taking me a minute to remember essentially like which book I had it kept in. I thought we learned about that in this class. Not seeing that. That's fine. You should be able to find it based off of the width of the river. Alright, so we're gonna go... I'm gonna skip some of these, like, I'm gonna skip some of these flies that you're probably never gonna see. Blepharocerides are only found at the bases of waterfalls. Deuterophlobiids are pretty much only found in, like, highly clean mountain streams. Crane flies. Those are guys you'll see. They have an end that has all of these kind of flanges. They can have all different shapes. They're big, they're fat, they are shredders. They are probably right there in the middle. They are not there's the word, moderately tolerant. Mosquitoes, obviously they are overall going to be tolerant to poor water quality. Mosquitoes don't really need oxygenated water, and they don't really need it to be clean at all. They can live in tree holes, they can live in the water inside of tires, and they can live in fetid, gross streams. Now, admittedly, you're not going to see mosquitoes in water that is moving quickly. Generally, you're only going to find them in the pools. But even in really clean water, you can find mosquitoes in the pools. Now, I want to share this with you now that we have mentioned that. I have something to share with you. So, if we're talking about, I'm going to give you a chart. And on this side, we're going to say diversity. And what I'm talking about is the number of individual species, like the number of species or the diversity in a river. And then on this, we're going to rank it essentially 0 to 10 polluted. Like the range of pollution. So, not polluted at all, absolutely pristine water, middle of the road, absolutely disgusting, gross water. Now, a lot of times, if you were thinking, people a lot of times think that this graph is going to look like this. This is what people think, that the less polluted it is, the more diversity you're going to have, and that's not exactly what we see. All right, so this is what's kind of in people's minds, but what is in reality, when you have absolutely perfectly clean, pristine water that is not polluted and it's perfect, perfect amount of oxygenated, your diversity is actually can be kind of low. And that's because some of our insects really kind of like at least a little bit of pollution or some organic material in the water or something. And so we find that with very, very little pollution, you can have a lower diversity. And then you end up with a chart that looks more like this, where the moderate streams are going to have the highest diversity, and then you can go off in either direction. And so this is something that I always thought was kind of interesting, and it's something that is, I think, important when we're talking about things like mosquitoes or things that are tolerant in polluted regions, because even though it's tolerant, it doesn't mean that's going to be the only thing you find there. It can be just one thing in a large amount. All right, midges, we generally, if you're going to consider them all as one family, you're going to consider them tolerant, because there are so many species of midges that are tolerant. If you have a midge specialist, there are some species that are sensitive and that you would be lucky to find, but those are really hard to identify. To identify a midge, you have to cut off its head and then mount its head on a slide and then look at it under a slide microscope. You also mount the body on a separate slide, or you can mount them kind of next to each other, but you mount midges to identify further than chironomedy, you have to mount them on slides. Just because all of the characteristics are kind of in their dentation in their head right here, and it's just this little itty bitty capsule. Oh, I hope to someday see your river again. Is your river kind of hiding? Why can't you see it? These are our black fly larvae that look like a bowling ball. I wanted to show you that image. These get these get a special call out. We did just draw a flower fly together, so we did just draw the adult, but this is a rat-tailed maggot, and they're generally considered a very, very tolerant species of absolutely disgusting water. And I would say if you find a rat-tailed maggot in your aquatic system, then you kind of can say that it's gross water. I think everybody else can live everywhere, but rat-tailed maggots actually need pretty gross water to survive. They have this really long tail, and at the end there's a spiracle so that our maggot can breathe above the surface. Oh. So a less pristine river might have more biodiversity. Exactly. Exactly, Avea. Alright, get to my next set. So I wanted to talk just a moment about mayflies, stoneflies, and caddisflies. They are very, very, very important in the grand scheme of everything. And we might as well just go to the Heptageneid. That's the one that I just showed you. I love his little face. All right. We did talk for a moment about our Heptageneid, or our mayfly, but what I didn't mention is that they are highly sensitive. In fact, what we're gonna say is we have an index, and I'm gonna just jump right into this one really quick. We have an index, and it's called the EPT index. All right, and this is going to, if your water quality is increasing over time, your EPT index should increase over time, and in a general rule of thumb. So sometimes the indices, they don't have an answer. Like, you can't get a number and say, all right, and then line it up to a chart, because every stream and every stream and river is different to itself, right? So go out to a river, go out to a stream, take your data and take your initial data, and then go out and collect again, and go out and collect again, and then see how these indices change over time. And that's really how they are most useful. So your EPT index is the number of species of mayflies, stoneflies, and caddisflies. The E stands for Ephemeroptera, and those are right here, our mayflies. Any of these insects with 3 tails. Not Jacques. Jacques is not a mayfly. Like this guy, 3 tails. And this one is a collector and gatherer, I believe. It could also maybe filter with those hairs, but not a predator. Anyway, you can see it has these 3 tails. So Ephemeroptera are the mayflies, and they have 3 tails. There's a huge variety and diversity of them, and you can dive in. Each one of the families has their own sensitivity ranking but you can call them all sensitive, pretty much. Betids are the ones that kind of break the rule, but that's okay. You can say you have 1 mayfly. All right. Now, the P stands for Plecoptera. Plecoptera are the stoneflies. Keep in mind, stoneflies, mayflies, caddisflies, they're all one word because they are not true flies. They are not in the order Diptera. Stoneflies have 2 tails. That's how you tell them apart. So mayflies and stoneflies are going to look very, 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 very similar. But mayflies have 3 tails, stoneflies have 2 tails. Sometimes mayflies' tails will break off, And you just have to be able to look really close at the base of the abdomen and see where the tails come out, and just make sure that it's 2 that it's a stonefly, or 3 that it's a mayfly. Oh, that's going to be so much fun. Oh, no. They clear cut all the way up to the bank of this creek. I hope they don't fertilize their lawn. A lot of that riparian vegetation is really good kind of filtration system for human stuff. You know, the plastic and all those types of things will get caught up in the riparian vegetation and not go into the water, and we can go and clean it up. Or any of our pesticides or fertilizers, things that people spray on their lawns, that vegetation will sometimes be kind of a natural filter so that it doesn't have to reach the stream. So it's kind of sad when people clear cut all the way to the creek. Yep, so those are stoneflies. There's all different types, and I guess that's them. Damselflies. We already saw a damselfly under the microscope. Also have 3, except, ooh, I want to show you this. That is what the bottom jaw looks like. So it has this shape that kind of looks like this. And that's the part that actually has the ability to shoot out and grab its food. So it has another piece that connects like this, and then it can straighten out. You see, like this. So it can come back and straighten out. And that's how it grabs onto its food. So damselflies are predatory. And dragonflies. And check out that bottom jaw. Yeah, there is a lot of really cool... Yeah, the jaws are really, really awesome. And there are a couple videos on YouTube of like slow motion of the dragonfly eating, of dragonflies eating things, or damselflies like catching mosquitoes out of the water. It's really the coolest thing. There are some dragonflies that get so large that they can eat like small tadpoles and small fish. And dragonflies. And this one is a funny kind of square bottom mouth. This is my favorite dragonfly in the whole world. It's the genus is Hey Genius. This is an odd dragonfly nymph. When you find them, it's kind of exciting because they're like the size of a... They're the size of like a... I'm trying to think of like a dollar coin. Like a Sacagawea. And they're flat, and they're round, and they're really adorable. They're just like little flat, round, coin-looking guys. And they're really smart, because their genus is Hey Genius. Yes, exactly. Totally different from a normal dragonfly. But they still have anal gills. They still have the ability to pull water up into the end of their abdomen, breathe from that water, and then if they get scared, they can shoot the water out of the back of their abdomen and use it like a jet propulsion. Alright, I'm going to say, as a general rule, we're just going to say this is a general rule, hemipterans, true bugs, are in that moderately tolerant range. There are some that are sensitive, but most of them are just kind of moderately tolerant. You've got these giant water bugs. We've talked a lot about those. Marley would have loved that. Yeah. Hey, genius. So we've talked a lot about giant water bugs. They're predators. They have venom. They breathe oxygen through a spiracle that breaks the surface. And so they are going to not need highly oxygenated waters. They also only really live in slow-moving environments because they aren't going to actively be swimming all over the place. They hang from vegetation on the edges. So you're more likely to see these in ponds and lakes or in the in regions of the water where they're slow moving. Yeah. Water boatmen have crazy little cute scoop-shaped tarsal segment up in the front. Back swimmers look like their color is upside down. They're very, very light on the top, and they're dark on the bottom, and that's because they swim upside down. So if you imagine why a fish is dark on the top and light on the bottom, it's because when you're looking down at the water, the dark matches the ground or the substrate, but if you're looking up at a fish, the light matches the sky, so it helps them kind of blend in. It's the same with back swimmers, except it's backwards because they swim on their back. Water striders. These guys run on top of the water. You should know those. This is another species of water strider that's a little smaller. It's got broad shoulders. These guys are adorable. We sketched one, the creeping water bug. They are like little itty-bitty miniature giant water bugs. They have piercing and sucking mouth parts and raptorial front claws just like them, but they're more likely found in quick-moving water because they do swim around. They're like the little itty-bitty mini version of a giant water bug. These are all such awesome notes. So you took all these notes in a college class. Yes. This was a class on, this was my, aquatic entomology class taught by Richard Merritt. He is the coolest aquatic entomologist out there. No, Ken and Marty would give me heck for saying that. All three of them wrote this book together. So I took this class with Rich Merritt, and he wrote the Aquatic Insects of North America book, and he was like the coolest dude. He was also a forensic entomologist, so shout out to Dr. Merritt. But I also, some of my other notes from the very beginning were from a different class. It was an in-person 3 weeks that we went to an environmental center, and I had the class with all three of them. Ken Cummins and Marty Berg also were there, and Marty is the one who does midges, and everyone gives him heck for studying midges because they're so small and no one wants to a slide mount their heads, but we laughed about it. These were, yeah, this, whenever I had an entomology class in university, I almost failed another class because all of my energy went towards studying entomology for 4 years. Like, I was the only thing I was doing. And, so it was like every time I had a semester with a really interesting entomology class, there was always one class that took the bullet. But I enjoyed myself. Yes, midges are the ones that fly around the heads, and they make those little clusters of flies, and they're no fun, and some of them bite, and, you know, Marty studies them. He used to be the head chironomid expert in North America. I don't know if he still is, or if he's finally retired. I think he was hoping to pass that flame on to somebody. They are dipterans. So, these are our midges. They're long and they've got these little bitty heads. They are a fly, and when they're adults, they look very, very similar to a mosquito. They even have that huge plumose antenna, just like a mosquito would have, except they don't have the long piercing mouthpart that a mosquito would have. They also, when they are standing, they stand on their front two legs and they hold their hind legs in the air. I wish I knew where my actual pencil went. I've got another one. So midges hold their legs like that. So their two front legs are down and their hind leg is up. But mosquitoes are going to be the opposite. The mosquitoes hold their back two legs down and they'll hold their front legs up. And so that's another way. If you see them land, you can tell them apart by just how they're standing. Rather the fly harms you. Exactly. I mean, mosquitoes are the most deadly insect on the planet. They cause the most deaths because of their transmission of diseases. And they're a true fly. And I'll admit, I've hurt quite a number of mosquitoes. I feel the same way. I feel like it's the dipterans that we should be concerned about. They spread disease. They are highly, highly adapted. I don't know. There are flies that, like the human bot fly, you know. There aren't any hymenopterans, bees or wasps, that can live in a human, you know. But there are flies that can live in people. Now, there are what we call the biting midges. Now, even though we call them midges, they are a different family, so you can go with that as you will. But they are long and thin, and they look like midges, except their front body, their pronotum, their first segment of their thorax, does not have any prolegs. Whereas our midge, I don't know if you can see that, but up really close, they have these little itty-bitty prolegs up there in the front, and biting midges don't. So that's how you tell them apart. Exactly. Oh, man. Yeah, so that's a little bit about those. I think we can skip those. All right, we're good. We'll probably skip that one. I've never collected a toad bug, and I want to because they are absolutely adorable looking, and they... I want them, but I haven't caught one yet, so we're not going to talk about them. All right. This is the final letter of our EPT index. All right, so we did E was Ephemeroptera for mayflies. P was Plecoptera for stoneflies. The T stands for Trichoptera, meaning the caddisflies. All right, the trichopterans are the caddisflies. Now, I would say out of the three, the trichopterans are the ones that have, and this is just in my experience, you probably could argue stoneflies too, but in my experience, the trichopterans are the ones that have kind of the widest variety of tolerance to sensitivity based on family. We do say that if you have more species of caddisflies that we generalize sometimes and we say that they are all sensitive. Some of them are moderately tolerant. So we'll go over, at least the one that I can think of is this one. This one is the moderately tolerant one. Pretty much all of the other ones you can consider sensitive in my head. So these are what we call the hydrocycids. They don't have a common name. Yes, they do. They are called... These are the net-spinning caddisflies. And I think... No. I don't have a specimen of one right now. I don't have a specimen of one right now, but a lot of times, at least the common ones that I see are regularly green. And I'm going to go ahead give us a little bit of a sketch. I got something to show ya. Once I get back to our page, I closed it again. Alright, so let's see. We're gonna go up here really quick. Alright, so generally the body of our caddisflies, of our net spinning caddisflies, are green. Caddisflies are kind of funny because they have these very bulbous, very soft, we would almost, not sclerotized abdomens, so they don't have a hard exoskeleton. Right here, it's kind of squishy. They also have these little, I guess I'm going to call them filaments, on the back of their abdomen. And then they have gills that run along the inside here. They look like they're kind of fluffy just on the bottom of their body. Alright, so those are all lots of little gills. Then you have the thoracic region, so you have 3 segments in the thorax. And then you have the head up in the front. And by the time you're up here, I believe they're mostly sclerotized, yeah. So, on our head, and then the regions in our thorax, a lot of times we have kind of these plates on the top, and those are what we call sclerotized. I'll give you that word. Oh, whoops. No! We're being eaten by net-spitting caddisflies. I don't like when I do that on accident because it messes up the format of my box. There we go. So we call that sclerotized is the word for hardened. Or to sclerotize means to harden your exoskeleton. So when we say an exoskeleton is sclerotized, it's just like it's a plate rather than it being squishy. So you can say it has a sclerotized head capsule. That would be using that word in a sentence. Or these would be considered sclerites because they are a piece of hardened exoskeleton. And you can see them here. When you look at them underneath a microscope, it's super obvious where our exoskeleton is really soft and squishy and where it's hard up here in the top, like little plates. Hardened head capsules. Yes, head capsules, and then they have them on the pronotum, the mesonotum, and the metanotum. So the first, the second, and the third segments of our thorax, they all have these little plates on the top. Depending on the caddisfly you're looking at, some of them have very small plates and some of them have larger plates. But all of our net spinning caddisflies have those. Now, they also are what we consider a moderately tolerant because you can find them, I think the net spinning caddisflies are the caddisflies that I see most regularly. They are found on rocks and they make these really adorable structures because if you noticed, there are no cases. And we always talked about caddisflies having and making their own cases. These guys actually just make a little net. They take 2 little branches or they can use a bunch of silk and they make themselves this cute little net on a rock. And then as the water is flowing by the rock and flowing through the net, that net is going to be picking up fine particulate organic material, the F-palm. It's going to pick up all of that organic material. And then the caddisfly, he's like, ah, yes, I have collected all of this food. I must now go consume it. And he'll go and he'll eat the entire net. He'll eat all of the organic material and the silk. And consuming the silk gives him the proteins back so that he can make more. Net spinning caddisflies. In the family Hydropsychidae. Alright. Now, I'm just going to go through really quick some of our caddisflies because I think they're adorable. And we're just going to look at the cases really quick so that you can see kind of what they look like. When you are sorting through a tray of insects, sometimes the caddisflies are a little bit trickier to find because they can just look like a pile of rocks, for instance. And you have to sit and watch and see if anything moves. This is what we consider a purse or a tortoise-like case because this one actually silks himself onto the rock, and he's got one flat side and one round side. So if we were going to actually look at him on a rock, if the rock was like this, he would be flat onto the rock and then over top of it like this. Almost looks like a little rocky scale. If you've ever seen like a scale on a tree, it almost looks like a little rocky scale on a rock. These guys, they make their cases solely out of very fine sand. These guys make their cases in little spirals like a snail shell. We call them Helicocycidae, but their body is even in this very round shape. So even once they come out of this shell, they are all like this little cute little C-shaped guy. And they do have this kind of little claw at the end to hold themselves into their case. All different, let's see. Aha! These guys are the ones that sometimes I have a hard time seeing because they look just like a tube of leaves. And if you have a whole big pile of leaves, seeing the tube of leaves can be a little bit difficult. And limnophilids are also pretty difficult sometimes because their stuff can vary. You can see it has some sticks, some leaves, some rocks, and they can really, really mix up their cases. These guys are fun because their homes are perfectly square. They cut all of these pieces of little stems in exactly the same length. And if you look at that case from down, if you like look down that case, he's like a square that gets narrow to another square. Yeah, so he's like this. Then you have some predatory. There are some predatory caddisflies that don't have cases. Yeah, so there's those. That was my exam. You don't need to see that. So those are stoneflies. I think that we made it through all of the most common aquatic insects. And we got to talk about mayflies and stoneflies and caddisflies. Woo-hoo! We're doing the things. Good. All right. Grand finale. Let's put it all together. Alright, this is going to be our actual field data sheet for collecting in the rivers. Now, we like to make sure that when you're going and you're collecting in the water body, because you've seen how many different types of animals there are. Let's see. Now you know, and you can see like how many different types they are and where they're found and their variety of jobs. It's important to know where you collected from. So we actually do record the habitat types. The percent cobble, which are would be stones on the substrate. The percentage of snags, meaning trees and fallen trees and clusters of leaves and those types of things. vegetated banks, percentage sand, those types of things. Now, they also generally will ask what you used to collect. I always prefer a D-net. It's just my preferred aquatic collecting gear. So, a D-net, you get this net and it looks like this. It is flat on the bottom and round on the top. And it has a kind of a short net that has mesh on the other side. And what you do is you hold that net so that the water is moving through it. You want to make sure you're always letting the current run through your net. And then when you are collecting, you want to be traveling against the current, because you want to go to as far if the current is going this way, you want to start down here and then work your way this way, so that when you're disturbing the water and when you're putting all of this dirt and stuff into the water and disturbing it, it's running away from you and you're not fighting your own mess. Okay, so you always work upstream. And then if I put a little person here, this person is going to be taking their feet and they're going to be kind of kicking the rocks and the substrate up and kind of towards the net. So as you're moving in this direction, you do what we call the macro shuffle. And so you shuffle your feet against the ground and you bring your net this way, and that's going to be your kind of preferred method for collecting along the bottom. You can also go up to the edges of the river, especially if the water body is eroding kind of underneath the bank. Sometimes taking this net and kind of putting it under the bank and swooshing up is going to get you a whole different variety of insects. So your goal is to collect from as many different habitat types as possible, and from as many different locations as possible along your 100-meter section, which is generally our rule of thumb for collecting. You pick 100 meters and you do that. They suggest to identify down to order in the field and then identify in a lab, but you can also just identify here. So this is going to be your data sheet. Your data sheet for identification. We call this the bench sheet because a lot of times this one is done after they actually collect. They put the specimens in alcohol and they send them to a lab to identify. And this is the form that they fill out. And it's the form that you can fill out. So you've got the organism, and you write down what order, and then as far down as you know, whether it's family, whether it's genus. You can also just count, like amphipods are scuds, so you can put down there, or our sow bugs. So you give as many organisms, you list them as possible. All right. You also want to know approximately how many. A lot of times with this, we do. It depends on whose form you're doing, whose index you're doing. But generally, if it's like more than 20, you can kind of stop counting at 20. You know, so if you found 4 or 5 dragonflies, where's Odonata? Oh, I guess Odonata it would be other, huh? If you found 4 different types of flies, for instance, you could put Diptera 4, or you could list like Chironomidae, Simuliidae, or midge, black fly, crane fly, those types of things. You give yourself a number. That's NO. LS means life stage, so whether it's a grub or an adult. TI is just your initials, and then the TCR is, we call it the, I always laugh about this, we call it the taxonomic certainty rating, which is just like 1 to 5, how certain are you that you identified that properly? because insects are difficult to identify sometimes, and even the specialists need to be like, I'm only 2 out of 5 certain that that's correct. You know, I laugh about it. All right. And then on the very bottom of our sheet, this data sheet gives us this series of indices. We can talk about a variety of other ones, but this one are the these are what this data sheet wants to record. So you've got total number of taxa, right? Obviously, if the number of taxa is going up, your water quality is going up. Hopefully, we learned that a little bit, that if it starts to go down, it could be because your water is super pristine. But as long as your number is going up, it's likely getting healthier and healthier. Your EPT taxa, that is how many mayflies, stoneflies, and caddisflies, how many different types you have found. You can give yourself a number, and if that's going up, you can say that your water quality is likely increasing. And then we have what we call the tolerance index. And the tolerance index is a whole nother calculation. So let's go into it. Now, the tolerance index is one that I like because you do end up with a number that you can say poor, fair, good, or excellent. All right, so this one, after you get your number, you can have a score or a rating for your stream. All right, your tolerance index, what you're going to do for this is you want all of your lists, you want your list of organisms, right? And you have divided them into those 3 categories we were talking about earlier. Sensitive, moderately tolerant, and then tolerant. So you divide them up into those 3. And I'll give you an example. So maybe we had 2 species of stoneflies that were sensitive. Maybe we had dragonflies. And maybe just 1. And then 1 species of damselfly. And then we had, I don't know, leech, which isn't an insect but is considered tolerant. Midge. 3 midges, and, I don't know, scud. Scuds are like freshwater shrimp. They're really cute. Okay, so we have all of these things, and this is all that we collected. Now, I might imagine that this might not be a super healthy stream, but we're going to go ahead and do the numbers. So what we're going to do, instead of just saying, I found 2 stoneflies. Let's see. We did say 2 different species, though. Okay. All right. So we have 2 here. The two different taxon that are sensitive. We have 2 different taxon here that are moderately tolerant, and then we have 5 that are tolerant. All right, we do weight these numbers differently. So the tolerant are only weighted by 1, so it's multiplied by 1. Our moderately tolerant are times 2, and then our sensitive, you multiply that times 3. All right, and this is not the number of individuals. So if I had 1 species of damselfly, but I had like 30 of them, you still just count it as 1. This isn't the number of individuals, it's the number of species, or the number of different types. All right. We summarize them all. We give them, we multiply them across. Then we would get what this is, 6, 4, and 5. And then we add those numbers together. And this would give us a rating of 15. Your rating can be anywhere from, well, I'll just give you what our answers are. So, if you have 10, we're going to say equal to or less than 10, we're going to call that poor. If you have 11 to 15, you can call that fair. If you have 16 to 21, you're going to be good. And then if you have 22 or more, you're going to call that excellent. And that is our tolerance index. When they say percentages, are they talking about percentage of stream covered by these elements? Yes, that is what they're talking about. So if we were looking at the percentage of cobble on the substrate, you're looking at the bottom of the substrate and you're saying what percentage, maybe it's 80% cobble, maybe it's only 15% cobble. Or how much percentage is covered by a log? Is it 10% covered by fallen trees, or is it like 80%? Is it completely covered by fallen trees that you can't move through? So that's what they're talking about. Snags, I generally are in the 10 to 25% range is where you're expected to see it. I always imagine cobble and sand next to each other. It's either like 75 and 25 or 50-50. Those are kind of substrate types. But they just want to give you, you're just trying to give information about it. So if we had a whole bunch more, if we had mayflies and stoneflies and caddisflies, our number could easily, very quickly get up to excellent. All right, does that one make sense to you? All right. So with our tolerance index, you determine sensitivity, moderately tolerant, or tolerant based off of their tolerance numbers that you can generally find in, you can find them in a couple of different books. You can find them in the Aquatic North America, the Insects of North America book. You can also find it in the EPA. You can also just, you probably could find them online in a variety of different research articles. All right, so there is, I was just trying to decide on which indices I really wanted to dive into for our final couple of thoughts, because we've been going at this for almost 2 hours now, and I know that there's a lot of information to consume. I'm so happy that we're doing this. So we talked about the tolerance index. We talked about the EPT index, which is important for watching those numbers go up or down. The number of individuals is important. And I was trying to pick another index that was maybe a little bit or slightly different than what we were talking about. So let's go into maybe habitat stability. There is a habitat stability index. All right, and you want your habitat habitat stability to be over 50%, or you want your answer to be over 0.5. So I'll go ahead and let's do that. All right, so habitat stability. For these types of indices, like if we were talking about riparian zone indices or habitat stability indices or even mobility indices, we're not talking about as much. We're not just counting how many different individuals are in the populations. We are now looking at what the jobs of those individuals are and how they are interacting with their environment. So with habitat stability, what you do is you take the number of filters and collectors, right? And you add those to the number of shredders, or the, let's see, give me a minute. Scrapers. Isn't it scrapers? Yeah. Sorry. These are filter collectors and scrapers. And then you divide that by the number of shredders plus the number of gatherers, collectors. So these are the filters, the ones that are going to need, let's see, the filters, they need a stable surface to grip onto a lot of times. Like you can imagine that bowling ball, the bowling ball, the black fly, it needs a rock, it needs a stable ground to be able to live. Our scrapers need a stable rock to be able to scrape all of that algae off of. So what you have here is the higher this number is, the more stable habitat you have in the environment. Shredders don't need a stable environment, they're going to be in like leaf packs. Gatherers are generally going to be wandering around the environment, so they're going to be going all over the place. They don't need a stable habitat. So when you're talking about habitat stability, or the percentage of stable habitat in your environment, you want this to be over 0.5, which is over 50%. Oh, good. I'm so sorry that your internet is going all wonky. Yep. So, I think that we've gotten enough. There are a couple more of indices, and if we really wanted to dive into it, you can always ask, and we can keep going, and we can go into more specifics or identify down into family of different animals. But I think for a 2-hour period, we did pretty good talking about general river habitats all the way down to collecting, how to collect, and how to come up with some numbers for yourself to determine water quality. So, let's see. Well, where'd the other page go? Gone forever. Oh, there it is. That's awesome. So we got to talk about habitat assessment. We got to talk about their jobs in the river, what those insects are actually doing. And there are probably 30 or 40 indices. You can determine how much sun is coming into the river. You can determine how much oxygen is in the water. You can do like predator indices. There are all types of stuff that you can do. But we got into the base level ones. We talked about the river continuum concept and how we look at rivers and how they grow and how the animals inside of their rivers change as the rivers get larger and the organic material that's passing down the rivers gets smaller and smaller. If you imagine yourself as a leaf, you might fall way down here, but then as you're traveling down this river, you might get cut up by a shredder, and then your smaller pieces, and then you might get picked up and eaten by a gatherer, and then you get pooped out in even smaller pieces, and then maybe a filterer will pick up all of what's left, right? So that's the thought behind the river continuum concept. We looked at the ID of a whole bunch of different aquatics. We talked about how to ID mayflies and stoneflies and caddisflies, which are the big ones. We talked about a variety of beetles and flies and butterflies that live in aquatic systems, and then did some math so that you could determine tolerance index and the water quality and habitat stability and all of those wonderful things. I'm happy. I hope you're happy. I hope that everyone has had a wonderful day today. Oops! Where am I? There I am. Okay. I actually never collected aquatics in this body, but there was a water scorpion on the top of it. I remember seeing that. I hope that everyone is having a wonderful day. Evea, I hope that you were able to hang around all the way to the end and that your internet stayed on. I had a great time talking about biomonitoring, and it's something that I have been passionate about since I was a little itty bitty person. I started volunteering for biomonitoring when I was 12 or 13. When I was 12 years old, I think is when I started volunteering. And then when I was 13, I started leading biomonitoring teams of people to streams to identify and collect and the like. So this is something that I have always been passionate about, but you can continue to learn about aquatic biomonitoring and aquatic systems for years and years and years and never know it all. All right. So having this 2-hour class, it gives you essentially a really, really, really great point to dive off of. And then if there are regions or areas of this that you'd like to ask questions about or that you would really like to dive into, we could take a whole nother class and just do the indices. But I didn't want to just go over the indices and how to collect without having you know why we're doing it and how to identify the insects we're looking at. All right. Thank you. I also teach on a platform called OutSchool. I teach to students ages 5 to 8, 9 to 12, weekly insect studies and junior bug club. I'm hoping to get my insect pinning class up and running. That one has been a little bit more difficult to get OutSchool to agree to for some reason. I'm not sure why yet. So that up there is a reminder to subscribe to my YouTube channel. All of you out there who have been viewing, if you've been in the chat box, I know you're subscribed, but there's a couple of you who don't talk with me, and I would love to hear from you someday if you would like to just speak up and say who you are and where you're from and how you heard about me. I love talking to new people too. And that is where you can send a couple of dollars if you really learned something from this class. If you are going to take information from this class and move forward and use it to biomonitor, please share that with me. I would absolutely love to hear from you. And if there were a number of you out there, this just occurred to me and I think it's really cool, so I'm just going to throw it out there. If there are a good number of you out there that are going to start biomonitoring, if you are going to start going into the rivers and going into the streams and start collecting and determining all this type of stuff, I might be able to create some Google Forms where you can actually hold all of your data. That would be kind of cool. I might design some Google Forms that would come up with these indices for you. That would be really cool. All right. Let me know if you want that. My email is trisha@theinsectopia.com, and you can always find me there. If you want to share your notes with me, you can go ahead. I know we didn't do an illustration today, but I hope that it was informative nonetheless, and I hope you all have a wonderful rest of your week. I will be back on Thursday to do a new scientific illustration, and have a great week. Stay buggy. Bye.